السلام عليكم. أنا الدكتور يوسف أحمد أبو ليلى، طالب الدراسات العليا بجامعة القاهرة، دبلومة الرقابة على الأجزية، وده presentation about electrophoresis. As this presentation we will talk about introduction, principles of electrophoresis, types of electrophoresis, application of electrophoresis, factors affecting electrophoresis conditions. Electrophoresis is a migration of charged particle in a medium under the influence of an applied electrical field. The usual purpose of carrying out electrophoretic experiments are to determine the number, amount, and the mobility of the component in a given sample or to separate them, to obtain information about the electrical double layer surrounding the particle. Principles: the existence of charge separation between the surface of a particle and the fluid immediately surrounding it. It's a process of separation, purification of protein, molecules, DNA or RNA that differ in charge, size, and conformation, migrate and the fluid around the particle to flow. Types of electrophoresis. The first type is a paper electrophoresis. In this, the sample is applied as a circular support on a strip of Wattman filter paper, moistened with the buffer solution. The ends of the paper are immersed in separate reservoirs containing buffer and in which the electrodes are fitted. By passing electric current, the ions in the sample migrates toward oppositely charged electrodes, and this method is suitable for separation of low molecular weight components such as amino acids and nucleotides. Applications of paper electrophoresis is the single most critical procedure in electrophoresis process. Special devices are available for this purpose. There is no hard and fast rule for the time of application of the sample to the electrophoretogram. This can be applied before the paper has been equilibrated with the buffer or after it. The second type of electrophoresis is starch gel electrophoresis. Starch gel are prepared by heating and then cooling a solution of partially hydrolyzed starch in an appropriate buffer. The separation is accomplished due to difference in charge as well as the size of molecules because of molecular sieving effect of the gel. Number 3, Agarose gel electrophoresis. Agarose gel are more porous and have large pore size and therefore used to fractionate large macromolecules such as nucleic acid. Agarose gel are cast by boiling agarose in presence of a buffer, then poured into a mold and allowed to harden to form a matrix. Higher the agarose, larger is the pore size. When the electric field is applied across the gel, negatively charged molecules move toward oppositely charged electrodes on the basis of their molecular size. Smaller DNA molecules will travel faster as compared to large one. Commonly used buffer are trace acetate, trace borate, or trace phosphate at a concentration of 50 millimole. Fourth type is polyacrylamide gel electrophoresis. The components used in this formation of the gel are known as neurotoxins. The most commonly used component to synthesize the matrix is acrylamide monomer, ammonium persulfate, and TEMD. These free radicals can activate acrylamide monomer, including them to react with other acrylamide monomer, forming long chains. These chains become cross-linked if the reaction is carried out of the presence of N,N-methylene bisacrylamide. TEMD acts as a catalyst of gel formation because of its ability to exist as a free radical form. The whole mixture then irradiated with ultraviolet light in the presence of oxygen. Buffer system gel electrophoresis can be carried out in buffer ranging from pH 3 to 10.2, and the ionic strength of the buffer system is generally kept low to reduce heat production depending upon the type of buffer system used. There is two types of polyacrylamide gel electrophoresis. Number 1, SDS polyacrylamide gel electrophoresis, reducing form, and the second type is SDS polyacrylamide gel electrophoresis, non-reducing form. SDS polyacrylamide gel electrophoresis non-reducing form. In this system, protein are dissociated into their subunit using ionic detergent such as sodium lauryl sulfate. The protein mixture is denaturated by heating the sample at 100 degrees Celsius in the presence of excess SDS. Under these conditions, most of polypeptide bind to SDS in the constant weight ratio as 1.4 gram of SDS per 1 gram of polypeptide chain. The intrinsic charge of polypeptide are insignificant as compared to negative charge provided by the bound detergent, and these migrate in polyacrylamide gel according to the size or molecular mass. SDS polyacrylamide gel electrophoresis reducing form. It's similar to SDS polyacrylamide gel electrophoresis described above, except that in addition to SDS. In this method, protein sample is boiled in presence of excess SDS and beta-mercaptoethanol to denaturate protein to their individual polypeptide. SDS polypeptide have net negative charge and migrate towards anode on the base of their size or molecular weight. And this is a photo shows us the difference between SDS polyacrylamide gel electrophoresis, reducing and non-reducing form. Applications of gel electrophoresis. This technique is used in molecular biology to determine of DNA sequence, southern and northern plotting, restriction mapping of DNA applications in protein study. Other types of electrophoresis. Isoelectric focusing. It's an electrophoretic method for separation of protein which differ in their isoelectric point. The proteins are made to migrate in pH gradient maintained by mixture of low molecular weight. Proteins migrate depending on their charge till they reach a region where the pH corresponds to respective isoelectric point, at which pH protein possesses no net charge and hence get focused. Applications of isoelectrofocusing. Electrofocusing has been used for separation and identification of serum protein. It's used by food and agriculture industries. It's used for forensic and human genetic laboratories. It's used for research in enzymology, immunology, and membrane biochemistry. Second type is immunoelectrophoresis. It's an Agaroz gel electrophoresis which exploits the separation of the protein on the basis of their charge to mass ratio and their antigenicity. Protein mixture are first separated by electrophoresis on Agaroz gel and then allowed to interact with specific antibody preparation. Antibody diffuse through the gel and forms visible precipitate with electrophoresed protein for which the antibody has affinity. The technique is used in determination of antigen concentration. As the antibody moves into the gel, it encounters antibody and precipitate when the antigen antibody concentration and equivalent resulting in the gel. Applications of immunoelectrophoresis. It's used to diagnose multiple myeloma. It's a disease affecting the bone marrow. It's also used to identify certain protein molecules produced by the immune system, and it's used to measure the Ig presence. Capillary electrophoresis. This technique that has very high resolving power and speed. Capillary electrophoresis involve electrophoresis of sample in small narrow capillary tube and high voltage from kilovolt is applied. This technique is used to separate biological material including amino acids, protein, nucleic acids, as well as drugs or even metals. Very small amount of this sample are required for analysis. Applications of capillary electrophoresis. It's used in the isolation of the correct optical isomer. Capillary electrophoresis plays an important role in ensuring a constant product quality of therapeutic glycoproteins. It also used in forensic area to detect performance enhancing drugs. The last type of electrophoresis is protein Western blotting. The transfer of proteins from the acrylamide gel into a more stable and immobilizing support, such as nitrocellulose filter, is called as protein blotting or Western blotting. Blotting of protein bands allow them to become more accessible for detection and identification using a variety of methods such as a glycoprotein, and also filter can be stored for much longer period. The transfer of protein from gel can be achieved by any of the methods such as capillary action, application, or application of vacuums. Factors affecting electrophoresis. Electric field. An electric field is property that describes the space that surrounding electrically charged particles, a movement of ions depending upon voltage, current, and resistance of the electric field. Number 2, the sample rate of migration is affected by the increase in size of molecule, inversely proportional. The third one is the buffer. Buffer affects migration rate of component and stabilize the pH of the supporting media. And there is two types of buffers generally required. Number 1 is loading buffer. This buffer be added to the sample of the PCR reaction that will be electrophoresed. This buffer contains a colorant like bromophenol blue that will allow you to visualize the sample when loading the gel and to track the migration material in the gel during electrophoresis. Number 2 is electrophoresis buffer. This is buffer covering the gel cathodes and anodes and contain appropriate ionic salt that will dissociate the travel across the Agaroz matrix with the movement of electrons from the cathodes to anode. Electrophoretic conditions. The conditions of electrophoresis depends on the strength of the current, the power applied, and the potential difference. These are dependent on the specific application, size of gel, and the percentage of Agaroz in the gel, density of the matrix. Most commonly, the criterion of 5 volts per 1 centimeter of the gel is applied and results in a total voltage of 60 to 100 for most gel. In general, higher voltage results in faster separation, but lower voltage permit most accurate separation and resolution of determination DNA species.